Good morning and good afternoon. Thank you for joining day 2 of our 2020 virtual user meeting, Membrane Physiology Beyond Ion Channels. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's presentations. By default, all audience members are muted until the presentation, at which time we would open the floor to questions. Please feel to address any questions throughout the course of the talks in the chat box. It is my pleasure to introduce Dr. Judith Stolwijk, Senior Scientist, University Hospital Regensburg in Germany, and furthermore, Michael Skiba, also from the University of Regensburg. Their presentations are entitled Impedance Measurements and Calcium Imaging to Investigate the Role of SORE-Operated Ca2+ Entry in GPCR-mediated Endothelial Barrier Regulations, and Profiling the Pharmacology of G-protein coupled receptors in cell-based assays using label-free impedance analysis. I will now hand over the presentation to Judith for the start of the talk.
Yes, hello everybody. I hope everybody can hear me. So welcome from my side as well to the Nanion user meeting. So I would like to present a project that I performed during my postdoc studies at the Colleges of Nanoscale Science and Engineering, and together with Applied Biophysics in Albany and Troy, New York. And this project involves using impedance measurements and calcium imaging as tools to investigate the physiology of some non-excitable cells, namely endothelial cells. And to be more precise, we were interested if the signal transduction mediated by G-protein coupled receptors involves the activation of so-called SORE-operated calcium entry. And we wanted to know if this mechanism of SORE-operated calcium entry is important to regulate GPCR-mediated changes in endothelial barrier function. So endothelial cells, you will know, they line all walls of all blood vessels, and they form an important barrier to prevent cells, solutes, and fluids to just leak into the tissue. But of course, in some cases, it is necessary that the endothelium can open up. So the endothelial barrier is under the tight control of some G-protein coupled receptors and their respective agonists. So for example, well-known pro-inflammatory agonists are thrombin and histamine that are known that when they bind to their respective GPCR, lead to an opening of the cell-to-cell junctions and to an opening of the barrier, and thus cells like immune cells and fluids can leak into the surrounding tissue in an event of inflammation. On the other hand, sphingosine 1-phosphate, for example, is an agonist which is known to be barrier protective. So when it binds to its respective GPCR, then the cell-to-cell junctions tighten up and the barrier function is enhanced. So how is calcium important in this pathway? Many of you will probably know that G-protein coupled receptors are intracellularly coupled to heterotrimeric G-proteins. And depending on the G-alpha subunit, there are different possible downstream pathways that are activated upon agonist binding. And especially the G-alpha Q and 11 family types are known to be activating phospholipase C. But note that also in some cases it has been described that the beta-gamma subunit can also activate phospholipase C. So phospholipase C cleaves the membrane lipid, liberating IP3, which can bind to the IP3 receptor in the ER membrane. And this leads to an opening of the calcium channel function of the IP3, so that now calcium, which is normally stored in the ER, enters the cytoplasm. So normally the cytoplasm is very low in calcium, and upon activation of the IP3 receptor, calcium is now leading to an increased calcium concentration in the cytoplasm, so that calcium-dependent proteins can carry out all kinds of downstream signaling functions involved. For example, many calcium-dependent proteins are known that lead to changes in the structure of the cytoskeleton and the composition of cell junctional proteins, which are all important to form the endothelial barrier function. So changes because dependent on certain calcium-dependent proteins can then lead to vascular leakage. To stop this kind of signals, calcium can be removed from the cell again, and this is carried out by different pumps, for example, the Circa pump in the ER membrane to pump calcium back into the ER, or also the PMCA at the plasma cell membrane. And especially the activity of the PMCA causes that over time the cell potentially depletes in calcium. And this has been a mystery for a long time, how cells can replenish their calcium. And it has about 15 years ago, a mechanism had been discovered, which is called store-operated calcium entry. This mechanism is carried out by the action of two types of proteins, STIM1, which is a calcium sensor located in the ER membrane, and ORI1, which is a calcium selective channel in the plasma membrane. And normally, when the ER is properly filled with calcium, calcium is bound to the STIM1 protein, and STIM1 is just distributed over the ER plasma membrane. But when the cell runs low in calcium and the stores have been depleted because of some signaling event by IP3, then the STIM1 molecules start to aggregate and move to areas close to the plasma membrane. There, STIM1 can interact with the ORI1 channels and activate them to open for calcium. So this mechanism now allows that calcium is refilled from the extracellular buffer. And especially for immune cells, but also for many other cell types, it has already been shown that this replenishing of calcium via store-operated calcium entry is very important to mediate proper signaling events. So obviously, this source of calcium is required for the intended signaling. So the question was now, if the store-operated calcium entry, if this source of calcium is also important in mediating changes in endothelial barrier function via the activation of different GPCR receptors. So that was the question. Is SOCE required for GPCR-mediated barrier regulation? And for this, we used human dermal microvascular endothelial cells to address this question. And we investigated the agonists that I mentioned before, thrombin and histamine, which are pro-inflammatory, and S1P, which is known as the anti-inflammatory agonists, to investigate how store-operated calcium entry is important in regulation of the barrier function upon these agonists. We used two methods. We used ratiometric calcium imaging in order to monitor calcium mobilization and impedance measurements to measure barrier function changes. So with ratiometric imaging, I guess many of you are well familiar. We use a calcium binding dye, Fura2, which is loaded into the cells, and upon excitation at 340 and 380 nanometer, we can measure the ratio of emission at these wavelengths. And you probably know that when the ratio of emission increases, that this indicates that we have a high calcium concentration inside the cytoplasm of the cells. Now, what we need to do in our case is that we need to distinguish two sources of calcium. We need to distinguish the calcium that is entering the cytoplasm from the ER stores and the calcium that is entering from the extracellular fluid via the ORI channels. So what we do is that we start out our measurement in a calcium-free buffer. So when we start the baseline and we add our agonist, we know that the calcium increase that we see here is coming from the ER stores because no calcium can enter from the extracellular fluid at this point. Then, of course, we have the activity of the PMCA setting in, which pumps out the calcium of the cell again. So this is the reason why we see a decline of the ratio. And at this time point, we have depleted our cell of calcium, and also the stores will be empty. So in the case we have activated our store-operated calcium entry mechanism via STIM1 and ORI, we will see a secondary phase of calcium entry as soon as we add back calcium to the extracellular fluid. And indeed, when we use our three different agonists, thrombin, histamine, and S1P, we see that we can measure both phases of calcium mobilization. The first calcium release phase and the second calcium entry phase. Okay, so this is how the calcium imaging traces typically look like. Now let's talk about how the impedance measurements work. So for the impedance measurements that are used to monitor the barrier function, I used the system developed and marketed by Applied Biophysics, Inc., which is known also as ECIS, which stands for Electric Cell Substrate Impedance Sensing. But you will see in the next talk given by Michael Skiba that also the cardio-excite equipment can be used to perform impedance measurements to study GPCR activation in other cell types. But what you essentially need is a coplanar electrode layout pattern onto the bottom of some cell culture ware, and you need an impedance analyzer. So you're able to inject a non-invasive alternating current to the electrodes, and then you can measure the impedance of the system. And the impedance is nothing else than to say, yeah, the resistance in an alternating current circuit. So when we grow the cells on the electrodes, these cells block the electrodes and prevent free current flow. So we have an additional resistance here. And the impedance will increase. So the method overall is sensitive, first of all, to changes in the amount of cells that cover the electrode, but the method is also sensitive to changes in the dimensions of these inter- and subcellular clefts. What you have to know about impedance, it's a complex number and it is frequency sensitive, and you can measure contributions that are coming from capacitive contributions, like for example, the cell membrane. and you can distinguish resistive contributions that are coming from the intercellular or subcellular clefts. And especially for measuring barrier function of endothelial cells, we want to focus on these intercellular clefts. So that is why many people, when they are studying endothelial barrier function with impedance measurements, use or focus on the resistance part of the impedance, which is typically measured at a frequency of 4 kilohertz. And that is why the resistance is denoted here. So when the dimensions of these clefts change, for example, when we open up here the barrier, the resistance of the cell layer, of course, goes down. And that is what we see in this corresponding impedance measurement curve. So when we have a look at the response profiles of our different GPCR agonists, we see for the pro-inflammatory agonist thrombin and histamine that indeed we see a decrease in the barrier function. For thrombin, it's a lot more pronounced than for histamine, but which also corresponds to the stainings that we can do on such cell layers challenged with thrombin or histamine, that we see for thrombin here, these intercellular gaps and a strong redistribution of the actin cytoskeleton. And also for histamine, we can see just not so pronounced little intercellular gaps which are open for current flow. And in contrast, for S1P, which is known to enhance the barrier function, the resistance go up, which also matches what we see in fluorescent stainings. Here you can see that the VE cadherin is enhanced at the borders and leads just to a stronger barrier of the endothelium. Okay, so we want to study how store-operated calcium entry is involved in these signaling events. So what we did now is we used certain store-operated calcium entry inhibitors, namely gadolinium ions or 2-APB, or we performed a molecular knockdown of the proteins involved. And to make things a bit more easier for you, I just focused on the results for histamine in this talk, but you can find the results for the other agonists as well in the corresponding publication. So let's see what effect gadolinium has on the mobilization of calcium. So gadolinium is a lanthanide ion which is known to clog the pores of calcium channels. And when we add gadolinium just in the beginning of our calcium imaging experiment, or when we add it just during the calcium entry phase, we see that gadolinium efficiently blocks calcium entry. So now we can use gadolinium in our impedance measurements. But what we could notice is that even when we shut down calcium entry via gadolinium ions, we still have this typical impedance response profile. So there was no significant effect of gadolinium ions on our histamine-induced barrier breakdown. Another inhibitor of store-operated calcium entry was 2-APB. The mechanism of action is not exactly known, but it is suggested that it somehow prevents the interaction between STIM1 and ORI1. And what we see in our calcium mobilization measurements, that 2-APB at a concentration of 25 micromolar inhibits calcium entry when we add it before the recording, and also it shuts down calcium entry when we add it during calcium entry. However, we need this high concentrations to really shut off calcium entry, because lower concentrations are known to have either no effect or rather can even enhance calcium entry. And when we use 2-APB in our endothelial barrier measurements, we could see that this immediate resistance drop that we normally see upon histamine stimulation is gone, is inhibited. So we could efficiently inhibit this typical histamine response. However, we see that later on, we also have a barrier decrease. And now one can think, okay, this is either a delay in the histamine response, but no, it is actually an effect of 2-APB itself. So 2-APB itself leads to a barrier disruption over time. And yeah, so this is an effect where you need to be very cautious when working with pharmacological inhibitors, that of course they can have some off-target effects or unknown side effects that you need to know about to be sure how to interpret your data. And therefore, often it is a more elegant way or an additional supporting way to not only use molecular pharmacological inhibitors, but to also do molecular knockdowns of the proteins involved. So in our case, we used siRNA to knock down ORI1, and we used these cells in our imaging experiments and could see that the knockdown of ORI1, of course, abrogates the store-operated calcium entry mechanism. When using these cells in impedance measurements, however, we saw that the change in resistance evoked by histamine is not altered with ORI1 knockdown. So this matches somehow to the data that we saw for blocking ORI1 with gadolinium, and indicates that calcium entry via ORI1 is not important to control these histamine-induced barrier changes. However, what we could measure is that when we seed the cells to the electrodes and monitor how the barrier function develops, so how the resistance develops of our intact endothelium, we could see that ORI1 seems to have an effect on this barrier maturation, because with ORI1 knockdown, the baseline resistance was significantly lower. Okay, we did the same now for STIM1. A knockdown of STIM1 also had the foreseen effect of calcium entry, namely that we completely inhibited calcium entry with STIM1 knockdown. Using these cells in our impedance measurements showed that this time we have an impact on the histamine-induced response profile. Namely, this transient resistance drop is not as pronounced as in the control cell layers. The good news is, however, that the STIM1 knockdown had no effect on the baseline resistance. This is important to know because we have seen that the level of baseline resistance has an impact on how strong the impedance drop gets. Okay, so in the end, what we could learn from these experiments is that the ORI1 channels are not important, obviously, for the GPCR-mediated barrier regulation, because we did not see an effect with neither gadolinium block or ORI1 knockdown. But it could have a potential role in de novo barrier formation of our endothelium. For STIM1, we noticed that it seems to have a role in GPCR-mediated barrier regulation, but obviously this action is not via the activation of ORI calcium entry channels, but likely via some other yet unknown effect. Okay, so I hope that with my talk I could show you how impedance measurements can be quite useful in the study even of non-excitable cells, and how they can also contribute to resolving effects of certain ion channels. Of course, I would like to thank all people involved in this project, my supervisors at CNSE and Applied Biophysics, and of course all sources of financial funding. And I want to thank you for your attention, and when there are any questions, feel free to ask me.
Thank you, Judith, for the presentation. Just in light of Michael's upcoming presentation, we'll take a handful of questions that have come in, and then any outstanding questions, Judith will still stick around to the end, and then we'll do another round of Q&A.
Okay, yeah, great.
There was an initial question: how relevant are GPCR receptors as drug targets? And as a second part of that, do you know how many of the modern drugs target GPCR?
Yeah, so GPCRs are very, very important drug targets. And it's about an estimated 30% of the drugs on the market that target GPCRs. Because, yeah, especially in the endothelium, they control vascular tone, blood pressure, inflammatory processes. So even alone in the endothelium, they are very important drug targets.
And another question regarding important benefits do you see in using impedance technology in GPCR research?
What benefits over a special other technique or overall?
I think overall.
So, yeah. So, yeah. So the benefits of impedance measurements is that you kind of get an impression of the overall response to potential GPCR agonists. So while other methods are focusing on one specific signaling pathway, like for example, calcium mobilization or CAMP-dependent signaling, many methods focus on just one of these pathways. But with impedance measurements, you can potentially detect all different GPCR coupling types. The only prerequisite is that the GPCR activation is somehow associated with a change in cell morphology so that you're able to detect it. Okay. And another benefit is that you don't need to work with recombinant cell lines. So you are not dependent on having a hexyl overexpressing a certain receptor or having a recombinant fluorescent detection system transfected into the cell, but the method is sensitive enough that you can work with, for instance, primary endothelial cells and really detect the signaling events in your relevant cell system.
Okay, perfect. Thank you, Judith. So now I kindly ask Michael to prep for his presentation, and then again, we'll address any outstanding questions that we've also received.
Yeah, great.
So maybe you could just share your screen now, Michael, and then you'll pull up the presentation.
Okay, can you see that?
Perfect, yep.
Okay, just a second. So, good morning, everybody, and thank you for the kind introduction before. In the next couple of minutes, I want to introduce to you the established impedance-based assays to profile GPCRs or G-protein coupled receptors. And in the end, I want to briefly touch a newly developed method to increase the throughput significantly of impedance-based profiling assays without loss in time resolution. To begin with, I want to give a short introduction about impedance measurements and GPCRs. I think we have heard a lot about it in Dr. Stolwijk's talk. So just a short summary. The impedance-based analysis of cells measures the impedance between two gold electrodes. So that's simply the resistance when alternating current is applied. And usually the working electrode is smaller than the counter electrode, which makes the cells seated on the working electrode the sensitive layer. And they are seated about 100 to 200 cells on the electrode. In most of my projects, I used the tabletop Cardio Excite 69 system from Nanion Technologies, which provides high throughput screening with 96 wells and fast data acquisition. And this picture here shows a typical array with the working electrode in the middle and the counter electrode around it. We've heard a lot about GPCR, so just again a short summary about the pathways that are coupled to the GPCRs. Dr. Stolwijk said already that GPCRs are among the most heavily addressed drug targets in pharmacology. I've heard about that 40%, so even 40% has been estimated of all prescription pharmaceuticals that address GPCRs. They are activated usually by a binding event extracellularly of a ligand, which induces a GPCR conformation change. And next, a G protein binds to the receptor, GDP is exchanged by GDP, and the activated G protein splits into a G beta gamma subunit and a G alpha subunit. The G beta gamma subunit can induce cellular responses. There are three main different G alpha subunits. In this case, the G alpha S subunit can activate the adenylate cyclase, which produces cAMP out of ATP. cAMP as a second messenger molecule can activate, for example, the protein kinase A, and then induce cellular responses as well. The G alpha I subunit activates in the opposite pathway, so it inhibits the AC, less cAMP is produced, and that's why less pKa is activated. This decrease of the cAMP level inside the cells induces different cellular responses. Last but not least, the G alpha Q subunit activates in the phospholipase C. We've heard that in Dr. Stolwijk's talk already. And this activation and the cleavage of this phosphatidylinositol biphosphate activates increases the intracellular calcium level. And with that, for example, in the PKC, the protein kinase C is activated, and again, cellular responses are, you can see cellular responses as well. There are several experimental approaches for GPCR profiling at different stages of the pathway. By using radioligand binding, the early phase of the GPCR activation, namely the binding event, can be observed. With, for example, the GTP gamma S assay, you can investigate, for example, agonism or antagonism activities. We've heard about second messenger molecules in Dr. Stolwijk's talk. It was calcium imaging. So the increase or the decrease of second messenger molecules can be studied as well. The holistic cellular response can be explored, for example, by gene reporter assays. But many of these profiling techniques are label-based endpoint assays. They are invasive and may require genetic engineered cells. In comparison, the impedance-based technology studies cell shape changes with non-invasive electrical signals without label, time-resolved, and even endogenously expressed receptor levels can be investigated. So now the established impedance-based GPCR profiling assays, and they can be classified, for example, into agonism and antagonism assays. And additionally, with the pathway deconvolution assay, the underlying pathway can be addressed. So this is an example of the agonism assay. In this case, it was performed with the Harcard cells endogenously expressing the human histamine 1 receptor, which is G-alpha-Q coupled. And after a baseline measurement here, we added histamine in different concentrations and recorded the change in impedance at 12 kilohertz relative to the point of addition. And what you can see is that the signal is dependent on the concentration of histamine. We extracted the impedance change at 8 minutes and plotted it against the concentration of histamine. And out of this concentration curve, you can get, for example, the EC50 value as a parameter to characterize this agonism assay here. The EC50 value is the concentration of the inflection point of the concentration response curve. And in this case, it was about 0.98 micromolar. The next example of an agonism assay was performed with the HEX cells artificially transfected with the human histamine 2 receptor, which is G-alpha-S coupled. So again, after a baseline measurement, we added histamine in different concentrations at time point 0. And relative to this point here, we recorded the impedance change at 12 kilohertz. As you can see again, the signal is dependent on the concentration of histamine, and we extracted the impedance change at 60 minutes, plotted it against the concentration of histamine as before, and the EC50 value in this case was about 0.7 micromolar. Last but not least, the G-alpha-I coupled pathway. In this case, it was performed, or the assay was performed with a CHO cell line artificially transfected with a human dopamine 2 receptor. There are two different dopamine 2 receptors, a short and a long splicing variant, and we used the long splicing variant. To enhance the signal of dopamine, a pre-stimulation of with forskolin was necessary. Forskolin is a direct activator of the adenylate cyclase, and it increases, therefore, the intracellular cAMP level. By blocking the AC, or the adenylate cyclase, by the dopamine-induced activation of the receptor, the decrease of cAMP was bigger and the signal was enhanced. Again, we extracted the change in impedance at 12 kilohertz relative to the point of addition at 40 minutes, plotted it against the concentration of dopamine again, and the EC50 value in this case was about 56 nanomolar. Just one example for the antagonism assay. In this case, again, it was performed with the HEX cells artificially transfected with the human histamine 2 receptor. And we used for this system cimetidine, which is a specific antagonist to the human histamine 2 receptor. We added cimetidine in different concentrations 50 minutes prior to the histamine addition in one single concentration. So 50 minutes after, we added histamine and recorded the change in impedance again relative to this point of addition. And as you can see, higher concentrations of cimetidine, for example here 100 micromolar in black, fully blocked the signal of histamine. And with decreasing concentration of cimetidine, the signal of histamine increased until the level of the control in light blue was reached. We extracted the change in impedance values at 60 minutes again, plotted it against the concentration of cimetidine in this case. And out of this concentration curve, you can get the IC50 value, which is the EC50 value for an antagonism system. And in this case, the IC50 value was about 5.1 micromolar. So next, the pathway deconvolution assay. This assay addresses the drawback of the impedance-based technology of not knowing how the signal is generated inside the cells. To obtain an insight on the ongoing intracellular processes, several model substances can be used. In this case, we used again the Harcard cells endogenously expressing the human histamine 1 receptor. And we added histamine in the control, which is only media. And we saw those impedance changes. And we wanted to know which pathway is coupled to this histamine 1 receptor, for example. The first substance which can be used was forskolin. As I mentioned, it activates in the adenylate cyclase directly and induces a CMP level increase. But in this case here, you can see that there's no impact on the signal, on the impedance signal due to forskolin addition. Next, calcimycin was used. Calcimycin is a mobile ion carrier for divalent ions like calcium. And as you can see, the signal decreased to the same value like the histamine signal before. So comprising the results, CMP level increase didn't affect the signal, which makes the G-alpha S or G-alpha I coupled pathway for the histamine addition here very unlikely. The calcimycin, which mimics a part of the G-alpha Q pathway, induced the same impedance change like the histamine addition. So presumably the H1 receptor activation in this case resulted in intracellular calcium level increase. With this assay, a hint to the intracellular process can be derived. Last but not least, I want to introduce to you a new method developed by Dr. Stolwijk and published from our lab in 2019. In this publication, a new method was developed to increase the throughput without loss of time resolution. This method of serial agonist addition is based on organ pharmacology. In a lot of those organ pharmacology assays, one organ is used to derive a full dose response curve. This method here transferred this idea to the cell-based assays. Usually, one concentration per well and cell layer is applied, which means a lot of wells and cell layers for a full concentration response curve. What I mean is that for 8 concentrations, you need at least 8 wells. To circumvent this limitation, the new assay used only one single well, and multiple concentrations were added successively with increasing concentration. After each addition, the signal was recorded. So here, for the comparison, the parallel agonist addition assay again. So this is the usual assay you have seen in my talk before. U373 cells were used, endogenously expressing the human histamine 1 receptor again. At time point 0, histamine was added in different concentrations, each in one well simultaneously. So this is in the usual graph with the different curves related to the different concentrations. The maximum impedance change of each curve was extracted and plotted against the concentration of histamine again. And the average EC50 value for that was 0.41 micromolar. In comparison, the new serial agonist addition assay. One single well was used and increasing concentrations were added successively. So first 0.03 micromolar, then 0.1 micromolar, and so on till 30 micromolar was reached. These concentrations here refer to the end concentrations, which were seen by the cells. The control was measured within an extra well. Each concentration was added 15 minutes after the prior one and calculated in a way that the respective concentration of each addition, for example, 30 micromolar, was applied. After the last concentration of 30 micromolar, an additional 15 minutes, the experiment was stopped. The maximum signal after each addition was extracted and plotted against the related concentration as before to obtain a full concentration response curve out of one single well with only one single cell layer. The mean value of the EC50 in this case was about 0.8 micromolar. So it was in the same range like the one of the parallel assay format that I've shown you before. To sum up, this method is very interesting when thinking about high throughput by keeping the assay time resolution as good as possible. With that, I want to take just a second to thank various people who helped me a lot with my project. First of all, I want to thank Professor Wegner for the supervising and Dr. Stolwijk for helping me when questions have appeared. Furthermore, I want to thank Nanion Technologies for providing the CE96 system, which was used to realize most of my measurements. And finally, I want to thank the GRK 1910, which has funded this project. Thank you for your attention, and please feel free to ask any questions.
Thank you, Michael, for the presentation. We had a couple of questions that came in. Earlier in the talk, you mentioned different cell lines. How does the cell line type influence the impedance measurements?
Okay. Well, we measured only the change in impedance, or we measured the absolute impedance, but we plotted only the change in impedance. But the absolute impedance is dependent on the cell line, so the barrier function, the distance between the electrodes and the cell layer, and so on. So you can see a difference dependent on the cell line in the absolute impedance values. Furthermore, we have seen that the same GPCR coupled pathway activation leads in different cell lines to different impedance profiles. So yes, the cell line does affect the impedance measurement. I'm not sure if I understand the question correctly.
I think that was it was just more an overall question. And then there was a question that came in both for Judith and Michael. So compared to label-based endpoint assays, you could potentially reuse the cells from the cardiac site in another experiment, and then did you ever try to do this, for example, RT-PCR or so?
Judith, do you want to reply?
Yeah. So yes, that is right. You can potentially use the cells for another assay, but you have to be then, of course, very precise with the timing. So you would have to stop the impedance measurement right when it's at its minimum or peak, and then you could, I don't know, isolate or fixate the cells and do staining of or qPCR. But it's really the challenge to have, when you want to do both assays, that in fact you would need another setup. So it would be nice to have a setup, for example, where you could do calcium imaging and impedance measurements at the same time, for example, or do some luminescence reading during your impedance measurements. There are some of these approaches around, but they are not so well developed yet that they are marketed by some company or so.
Okay, and then we had another question come in. Could you say how this barrier breakdown can be explained in diseased cell lines as in cancer cell lines? And I guess maybe both of you can comment on that.
Yeah, sure. So when you have, so you mean probably that when you have cancer cell lines, you have not like the classical barrier breakdown, but what you have is some morphology changes, for example. So when the cytoskeleton inside the cell changes due to some signaling event, maybe the cells shrink a bit or they start to flatten out on the substrate. This also translates into changes of the subcellular and intercellular clefts, which then changes the impedance. And of course, with the endothelial cells, because it's really this barrier formed by the cell-cell junctions, which is so an important factor in endothelial barrier biology, that it's just why people specifically call it barrier function and not simple impedance change. If this answers the question.
Okay, thank you. So what I'll do is I'll make sure that this whole presentation is available on demand in case there were some individuals, obviously due to the time difference in North America, it could be a little difficult. I'll make sure to link both the contact information for Michael and Judith. And again, you're welcome to reach out to them directly. We're also able to answer any questions you might have regarding the systems used in this talk. And I have a kind reminder that at noon, we're actually opening up a small little quiz that we have on our day 2 proceedings. And this essentially will be a live raffle for a nice little, reward that we're sending out to all of our participants. So that's up on the website already. And our next presentation will begin at 4 p.m. Central European Time, 10 a.m. on the East Coast. Dr. Matthias Quick from Columbia University. Thank you so much for attending. Enjoy the rest of your morning.